Okay, this is just going to be a quick video going over the project in a little more detail and depth than what we've been able to talk about in the past few meetings. I just kind of wanted to keep the ball rolling since we did have to miss the past couple weeks. So while you're listening, just be thinking of any questions you might have. I'm going to go over some of the background of the project as well as some of the data that we've collected so far. So, like I said, just be thinking of any questions and any connections you might want to make based on what we have already. So just kind of jumping in, the population that we work with are individuals with hemiplegic cerebral palsy or CP. And so these are individuals who have had an injury to their brain either before, during, or shortly after birth. And the reason that we term it hemiparetic or hemiplegic CP is that one side of the body is affected while the other is relatively unaffected. So you can kind of see in the image, there's some different subsets of CP, and hemiplegia is the one where it's really just one arm and one leg, potentially both, sometimes one or the other that's affected, and that's because only one side of the brain is injured, either due to a stroke, some sort of malformation. Really, just that brain tissue can be very vulnerable during that part of development. And so, one side of the brain in individuals with hemiplegic CP has been affected, and so the opposite side of the body is affected. Um, you can see there's other types of CP. You'll learn a lot more about this in lifespan in the fall, but it really does correlate to the part of the brain or the structure in the brain that was affected by the injury. And kind of what we're trying to investigate is what's going on in both the injured side of the brain as well as the uninjured side, and how that affects the different motor deficits, motor impairments that we see in this population. So it affects movement and muscle coordination. A lot of times these kiddos are diagnosed because they have delays in their motor milestones, like reaching, crawling, walking. And a lot of times they don't even get an MRI as part of their like clinical care. So that's kind of a benefit to them participating with us. They get an MRI, they can take it to their neurologist, but CP is diagnosed just based on motor deficits. So often they are just kind of seeing like, oh, one side of the body is affected, both sides of the body are affected, all four limbs, or for example, in diplegia, the legs are often affected more than the arms. And even physical therapists, a lot of times pediatricians, are the ones using that information to diagnose cerebral palsy and the different subsets. But specifically, hemiplegic cerebral palsy, like I said, the arm and the leg on one side of the body, opposite to the side of the brain that was injured, is affected. And what we see is that there's a really wide spectrum of deficits. So, just anecdotally, I've had participants come in who talk about how they participate in sports, they play instruments, all of those sorts of things. They interact with their peers normally, and they may have like a bit of a deficit in terms of how strong they are, but otherwise, you wouldn't really even know that they have CP. Other kiddos who come in, they find it difficult to grab and pick up objects. A lot of times they may even have difficulty walking short distances without their AFOs or other orthoses. So it really just depends on the kiddo, on their, medical history and things. But what we're interested in is seeing if there's anything in the structure of the brain that can tell us when these deficits are going to be worse than others, and how that can lead us to really individualized treatment for these kiddos in the future. Um, and one thing that I guess I forgot to mention, and that we're kind of moving towards, is that CP is a lifelong disorder. It's diagnosed in children and it's typically thought of as a childhood disorder, but people live with this. It's not progressive, so the brain injury isn't going to get worse. We do see secondary musculoskeletal changes like contractures, and muscle atrophy. And so there are adults living with CP who have a lot of pain, a lot of difficulty doing different types of dexterous movements and things. And so just something to keep in mind when we're talking about CP, it's not necessarily just a childhood disorder. It's also something that adults have as well, and adults go to physical therapy for. So yeah, let me just go to the next slide here. So we're really interested in the upper extremity in kids and adults with hemiplegic CP. And really a lot of past work has focused on the shoulder and the elbow. And so we're kind of working our way down the arm. And now we're looking at motor deficits in the hand. And so two big ones that are really characteristic for CP are weakness and mirror movements. So weakness, kind of like I mentioned, some kiddos don't really have that. Like they're able to hold objects, pick up objects without really any issues. Other kiddos can't pick up a glass of water. They can't really manipulate different things. So they have difficulty typing, tying their shoes, carrying different objects if it requires two hands. So we're interested in how the affected hand's weakness compares to how strong their unaffected hand is. Pretty easy to wrap your mind around, just grasp weakness. The other thing that we're interested in is mirror movements. And so this is something you may not have heard of. I know we talked a little bit about it, but basically what happens is when you ask some of these kiddos and adults to do a certain movement with one hand, they involuntarily do it with the other hand. Even if you ask them to suppress it, they may not be able to. And that's just a result of the condition, as well as the motor pathways in the brain and how they're reorganized based on the injury. So this video is an example. I'll play it in just a second, but basically what you're going to see is I'm asking the participant to open and close their right hand. And so their left hand is kind of resting on their knee, and they're able to open and close their right hand really well. But their left hand is also doing the same exact motion, not necessarily at the same amplitude or intensity or magnitude, I guess. But then they're not able to suppress that movement. It's just something that happens naturally when they try and do it with one hand. So I'll go ahead and hopefully play this. Okay, so yeah, you can see he's opening, closing, and then both hands are along for the ride, without really him being able to control that. And so not to get into the weeds too much, and I'll kind of move myself there. But what happens in development that causes this is based on the motor pathways and how they're pruned in typical development versus how the body tries to compensate when there's an injury. So in typical development, everyone, as they're in utero, they have both contralateral and ipsilateral motor pathways. So in the image on the right, you can see the ipsilateral ones in purple, contralateral in blue and red. So what happens as after you're born, the blue and the red are strengthened, and that's how we have the opposite side of the brain controlling the opposite side of our body. And that's just a result of normal development. You no longer need those ipsilateral tracts, and so they're pruned. The body naturally gets rid of them. The use it or lose it phenomenon. So the body really just uses that contralateral opposite side motor pathway. And so the purple ones go away, blue and red are strengthened, and like I said, opposite side of the brain controls the opposite hand. But what happens if there's an injury early in development? So you can see here that red is kind of just signifying an injury. That may be a stroke, that may be an enlarged ventricle pushing on the motor pathways during development. What can happen is now the body does naturally get rid of that purple pathway, that ipsilateral pathway. It doesn't need it. But then it also no longer has that blue pathway leading to the opposite hand. So instead, it uses the resources it still has, and both the red and the purple are strengthened as much as possible. And so we think that this is the basis of those mirror movements, is that there's a motor command that's originating in that uninjured hemisphere, And so when you're trying to do a movement with one hand, you're automatically doing it with the other because of the entanglement of those pathways. And then what we're also trying to figure out is if you still have some of that blue pathway that's contributing to the strength of that affected hand, maybe you don't do as much mirroring, and how that all kind of plays out in motor impairments and what we can find out from the brain with that. Okay, so that was kind of a lot. Maybe it might have generated some questions. I know I probably didn't explain it like super clearly, but hopefully it's making a little bit of sense in terms of background. The next couple things I'll go into are the methods of what we collect in terms of the imaging. So I'm gonna try and switch to my iPad here, just so I can draw out a couple things. Let's see. Okay, so this is a structural image of a brain. So this is a pretty typical MRI that someone would get if their neurologist ordered an MRI and they just needed to see structures of the brain. This is an individual with CP, and you can see, I'll kind of circle it here, this ventricle is enlarged. And so that would be the side of the brain that we consider to be affected. And what we do with our MRI process is we're trying to look at the structure of the pathways of the brain, where they're leading, how much of it there is, and how it compares from side to side. And the way that we do that is through diffusion imaging. And what we're looking for is the diffusion of water. When I say what we're looking for, basically it's just how the machine, how the MRI works and the gradients that are used that picks up the diffusion of water. But really what you need to know, so if we think about the ventricles here, this is more or less free water. So if you have a water molecule, it can move in any direction freely, like it's not going to be restricted in any way. So this part of the brain is going to have a very low fractional anisotropy. That's the FA value that you probably saw if you looked at the IRB, if you looked at the paper that I sent. That's kind of the value that people use when they're looking at diffusion MRI. And that's just a value that tells you the probability that water is going to move in a certain direction. So with free water, FA is 0 because it's can go in any direction, and it doesn't have any preference. So when you think about that, maybe like graphically, in an image, it's just a sphere because that water can go anywhere. So in any way, three-dimensionally. That's how we think about cerebrospinal fluid in the ventricles, surrounding the brain, all of those things. Then next, you can think about the gray matter. So that's all surrounding the brain here. And this is a T1 image, so it shows up gray. So in gray matter, your water molecules are going to be a little bit restricted by different types of cells, brain cells, astrocytes, glial cells, all of those things. And so it can still move really in a lot of different directions, but it might just be restricted in some more than others. So that FA is actually going to be a little bit higher. It's not going to be 0, but it's not going to be close to 1 either. So, I mean, it can be really anything from like 0.1, 0.3, those sorts of values depending on the properties of the tissue. And what that's going to lead to is you can think about it graphically as more of like a football shape. And so instead of this sphere that we have here, we're going to have something that looks a little more ovally with one maybe preferential direction that way, and then the other directions It can still move in them, but maybe not as much. So then the last, I guess, extreme is when you have your white matter pathways. And so that's all of these pathways that are running through the brain here, like this. And what happens here is that you have water molecules, but all of these pathways are axons. And so you have all of your axons running like this. And so your water molecules really only have one direction to go, up and down. They can maybe go, because they're smaller than the axons, they can go kind of side to side within them. But the reality is they're going to be moving a lot more unidirectionally. And so those FA values are going to jump up even higher to 0.4, even up to 0.6, 0.7, depending on how restricted they are in their movement. So now you're going to get an even skinnier oval or ellipse, and you're going to have a stronger preferential direction. So this was like a really basic explanation of FA and what diffusion tensor imaging or diffusion weighted imaging is showing us. So I can talk a little bit more about that on Monday, but basically, this is what we're using to determine how the different sides of the brain are affected by this injury. And then we're using this and comparing it to the motor impairments, which I'll talk about in a little bit. So now I'm just going to go back to presentation. Keep going a little bit. Okay, so here are some examples of the images that we get, and then using those FA values, we're able to isolate the corticospinal tract. That requires a little bit of post-processing where I actually kind of color in basically different structures in the brain, the cerebral peduncle, and then the posterior limb of the internal capsule. Those are two really good landmarks for finding the corticospinal tract. And then the software algorithm connects them and then drives that connection up to the cortex as well. So you can see in these images, it's just kind of giving an example of a moderately impaired individual, and you can see they obviously had this hemisphere of their brain affected pretty significantly by ventricle enlargement. And so in the red, you have their unaffected corticospinal tract, and in the blue, they're affected. And so you can kind of see this is just one slice. If I were to cut down the front of the brain like this, and then that blue and that red will change based on whether you're going forward or backward. And I'll show that a little bit with the software as well. In the middle, you see more of a mildly impaired. If you were to show this brain to a radiologist, they may not even know that this individual has cerebral palsy. And so we can kind of use diffusion tensor imaging a little bit to dive deeper into the brain structure. And then on the right, you have a typically developing individual. You can see there isn't really a whole lot of difference between the middle and the right, but we're still able to capture some differences when we look at the corticospinal tract specifically. And then here is just kind of that idea that we're just looking at slices of the brain. And so that corticospinal tract is really three-dimensional. And so I can kind of show that here. So going from back to front, you're seeing the white is the corticospinal tract. All of the different colors have to do with the directionality of the fibers. So not super important right now, but just kind of showing we are really looking at this as a three-dimensional structure. And then in terms of the hand impairments, so when we have a kiddo come in, we do the imaging, which for them is really passive. They're just laying there. These images don't require them to be doing anything while they're in the scanner. They're just watching a movie or listening to music, depending on what they choose. When we want to understand the hand impairment severity, they're playing this almost like red light, green light game. So they have two dynamometers. These measure forces that they're squeezing, isometric, so it doesn't move. They're just squeezing and they'll measure force. And it's delivering that information to my laptop. But then when the light turns green, they're squeezing as hard as they can. And then when the light turns red, they're stopping. So in this example on the screen right here, this individual was asked to squeeze as hard as they can with their left hand, and the right hand is a red light. So they're not volitionally or voluntarily squeezing that right hand. They are doing so as a result of mirror movements. So I'll play the video, and you can see in the corner over here, that's their force output. So they're giving a max force in red with their left hand. And then their right hand, even though it doesn't seem like it's a lot of force, you can see it follows what's happening with the left hand. And then because that is their affected hand, it's actually a really high percentage of the force they're able to produce. So because we're looking at both grasp weakness and mirror movements, we can use those together to figure out how much they are mirroring in terms of what their max force is in their affected hand. Along with this, I feel like my laptop's about to take off. Along with this, we also do clinical assessments. So you probably haven't learned yet about the Fugl-Meyer, but that is one that's typically used in adult stroke. And so we can look at upper extremity function, and then it gets really granular all the way down to wrist and hand function. So we can use those measures as well to classify individuals based on their severity. And then the other one that we do is the TASC, the test of arm selective control. And that's looking at how well they can isolate joint movements. So they do shoulder flexion, shoulder abduction, elbow flexion, wrist flexion, and then a couple different types of grasps. So pull hand, pincher, and then key grip. And that one is really nice because it does capture mirror movements. That's one of the criteria for scoring. So I can go into those a little bit more in depth on Monday if that's something that you want to know more about. But basically that is an overview of all the data that we have from the project so far. We have imaging data, which gives us an idea of those pathways and how they're affected in each hemisphere of the brain. We have this grasp strength data and mirror movement data that I've kind of gone through and given a severity for. And, you know, higher numbers, we've decided to make it so that the higher the number is, the more severe impairment it is. So everyone is basically given a number based on how they're performed in this like red light, green light task. Then we have the clinical assessment, Fugl-Meyer, and task. And that kind of is what we have for each individual. Some people we have better data than others. Some people have missing data. That's just the reality of research. But altogether, we've got, I think, 35 participants, around half of which are individuals with hemiplegic CP, and half are typically developing kiddos and young adults. We do have some adult data. It's not as fleshed out as our kiddos and young adults, but something that we're looking to get into a little bit more. Before I forget, one thing that I also wanted to show, so the other piece of data that we have, I'll share my screen again, has to do with structural imaging and the different structures that we can parcel out in the brain. So I put an example of this in the synthesis folder that I shared with you all. So what I want you to try to do, just to make sure that your software is working pretty well, is to open up this image viewing software. So if you just search for it here, and then it's here in applications. And now that I'm saying that, I don't think you can see it because I'm sharing just my screen, but command space and then search FSL EYES, and it's that application. And then this will come up and you'll hit the plus button here. It'll take you to what's like the directory for the software. But if you have OneDrive on your computer, which I think is super helpful, or if you downloaded them, that's fine too. If you go to the synthesis folder and then images, and then you can open both of these right here. And then they'll come up and it's gonna look like this at first. That's because the one on top is a label volume and the one on the bottom is an actual image. So what I do now is I have this labels selected and then I go up to this top toolbar up here and I change it to a label image. And that's going to turn all of those like gray, white, black colors into like colors that I can kind of look for more patterns more easily. And as you can see, they line up with different structures within the brain. And so I luckily have a lookup table that shows me each of the you can see down in the corner, sorry, down here that shows each label value. And then I went in and typed in every single name of the structure as well. But also in that folder, I have a Excel sheet and a Word document, I believe, or no, like a PDF picture, where you can go and you can see which number goes to which abbreviation and then which abbreviation. You can look that up if you need to, in the PDF. I'll try to make something better. I tried to figure out where the file was for this like number to name thing in the software is, but it's eluding me. So I'll work on that as much as I can. But basically what you can see now in this image is you can click and it's going to tell you down in the corner at the very least the number that corresponds to that structure. And so I can see here, this is the superior corona radiata in the left hemisphere. And you can see that even though it's on the right, it is the left. That's just radiographic convention. So left hemisphere is on the right side of the screen, right hemisphere is on the left side of the screen. But yeah, I can click around. I can see this is the middle frontal gyrus. Over here, the precentral gyrus that we know is the motor cortex on the right is down here. And so I've used this previously to look at different correlations between the corticospinal tract and where it's terminating in the cortex in kids with CP versus typically developing kids. So that's something we can explore further. Just another piece of data that we have, luckily, from the imaging. And then, yeah, if you've never seen a brain MRI before, here's what a structural image looks like. So it's really nice for looking at structures. You can see again, here is a malformation in the ventricles, so an enlarged ventricle over here. I believe this is the data from the individual that was in the videos that I showed in the slides. Not 100% sure on that, but I think it is. So you can see here, their left side was affected, which means that their right arm and leg are the ones that have the motor deficits. But yeah, I feel like at this point I am rambling. So hopefully this generated some questions, and we can talk about this more on Monday. I definitely could have made this like an hour long with how much background there is for methods and things. But really, I just want you all to get a good picture of what we have going for us right now, and then we can use that to kind of talk about next steps and what we want to do with all of this great data. So yeah, email me with questions, and I will see you all on Monday.